of PopBio on the dark side. Today, it's my pleasure to introduce Dr. Andrew Barnes, who's a senior lecturer and head of the EcoDiv research laboratory at the University of Waikato in the north central part of the North Island of New Zealand. So I think we're meeting the goal of expanding our geographic scope of this seminar series all the time, which is really nice. Previously, well, Andrew was actually telling me a little bit before that he doesn't know where home is or where he's from. But at least academically, he did his Master of Science at the University of Canterbury in Christchurch in the South Island of New Zealand, before doing a PhD at the University of Göttingen in Germany. That's in Göttingen, Andrew, is that right?
Yep.
Yeah.
Yeah, sort of right in the center of Germany.
Right in the center. Okay. So Andrew has a number of interrelated research interests. They mostly focus on the drivers of ecosystem function, particularly the role of biodiversity driving ecosystem function. So this is a long-standing scientific problem in ecology, a really interesting one that throws up new challenges all the time. And I think Andrew's bringing a really interesting lens to this problem. He's looking at the contribution of biodiversity in complex ecological communities, so typically involving multitrophic interactions, through the lens of energy fluxes between links within these ecological communities. And so that's something that Andrew's developed a reputation in since sort of the mid 2010s to 2020, like around, I don't know, sort of emerged over a long time. And I believe that's what he'll be talking to us about today. I think another really interesting thing about Andrew's background is that he's been very consistent on his interest on ecosystem functioning, but has not restricted is developing new approaches, which he'll talk about today, but has also tackled this problem across a really wide range of scales, from small experimental plots right up to whole landscapes in the tropics, for example. So really sort of tackling this problem in all sorts of really interesting and important ways. So really happy to have you here, Andrew. Really looking forward to your seminar. Go easy on Andrew. He's literally just finished his teaching obligations for the semester. So yeah, but we all look forward to your talk, Andrew. Welcome.
Right. See if we can share the right screen here. Thanks.
Yeah, don't share the wrong one.
Classic mistake. No, I've got the wrong one there.
That looks good.
Oh, sorry. Hang on. Apologies, just bear with me. I'll swap that around. All right, hopefully that's...
Yep, that looks good, Andrew. Thank you.
Excellent. Great. Thanks very much, Simon, for that shining, very positive introduction. I should ask you to write my sort of profile statement on my next promotion application. Yeah, I guess today what I'd like to do is try to give a reasonably short, hopefully, overview of, I guess, some of the main things that's kept me really interested in research over the last, yeah, geez, I suppose decade, nearing a decade now. It's kind of scary when I say it like that, but yeah, I guess you know, from kind of early on, I became quite interested in this field of research of trying to understand the linkage between biodiversity and ecosystem functioning. But as I suppose, you know, started with kind of an early interest in animal ecology, particularly in invertebrate ecology. And so it always bothered me a bit that, you know, much of this research was focused on sort of single trophic level of primary producers. So that's, I guess, kind of where things took off for me. So yeah, I just want to start as well by generally saying, you know, thanks very much to the organizers of this seminar series. It's an awesome idea, and it's yeah, feel quite honored to be presenting in it. So yeah, and really looking forward to the talks coming in the future as well. So I thought it would probably be good for me to just start off with a bit of a disclaimer that you know, I'm presenting here in a theoretical ecology seminar series, but I would normally sort of describe myself as someone awkwardly sandwiched between empiricism and theory. I do have a strong interest in theory, but I would argue I'm probably not so good at it. I tend to like to sort of take theory and see how I can apply it to empirical systems. So, you'll probably be spared of too many equations and that sort of side of things, but much more focused on a couple of case studies as well of how we're trying to kind of integrate biodiversity ecosystem functioning research into multitrophic systems. So I think it's probably worth starting off just kind of setting the scene for, I suppose, my motivation for this topic. So as many of you will probably be aware, we're in a sort of a biodiversity crisis. It's been named a few different things, but I suppose the evidence for the so-called biodiversity crisis largely stems from the sort of global indices, such as the Living Planet Index, where we can see that the average population size of vertebrates has been decreasing pretty steadily since, in this case, since the 1970s, from when this index began. And we also know that, on average, extinction rates are much higher than background rates and, been steadily increasing for the better part of a few hundred years. So, I guess then that leads to the question of what evidence do we really have for biodiversity loss at a local scale, which is the scale, essentially that ecosystem functioning is kind of relevant across the planet. And some of you might be aware there's actually quite a large debate about this apparent or I guess purported decline in global biodiversity. Because in actual fact, we do not have very solid evidence for a steady decline across the planet. In fact, it looks a bit more like this, where we have kind of a mix of, some regions of the world are clearly declining over time. So this is taken from a big global meta-analysis of time series of biodiversity data. And what they've shown is that, some parts of the world are steadily declining. Other parts of the world seem to be steadily increasing in diversity. And, I'm sure many of you can guess why that is, particularly anyone that's familiar with the situation in New Zealand. We're gaining lots of species, but, this is largely through species introductions. So, what we have is more of a story of biodiversity change in terms of changing composition of species as opposed to necessarily a net loss, throughout at a local scale throughout the globe. So ultimately, biodiversity changes is a bit more complex than simply a loss of diversity per se. I guess probably also worth pointing out that I suppose there's a few different reasons why we might even care about biodiversity. Many people have asked me, why should we even care if biodiversity is declining or increasing or changing? Certainly there's some inherent sort of value to biodiversity that's hard to describe. It's just the variety of life, and humans are just fascinated by diversity, I would say by and large. But also, I guess suppose in more recent times, in the last few decades, sort of early 90s, David Tillman, namely, sort of flipped this idea on its head that ultimately ecosystem processes drive diversity, to rather ask the question of how diversity in fact drives ecosystem functioning. And so, since this famous experiment, the Cedar Creek or Big Bio experiment in the northern US, since that began, we've really amassed decades of evidence that there is, in fact, a positive influence or a causal linkage between species richness or actually different facets of diversity and ecosystem functioning. And it really all comes down to this sort of early evidence that made quite a big splash. Ultimately, increasing species richness leads to increasing biomass production of plant communities. I'm sure there's a lot of variability around that, depending on the systems you look at, but by and large, that's the general trend we tend to see. However, as I've kind of alluded to, much of this evidence really comes from plants, and the vast majority is focused on single trophic levels, and that's, I would say, largely because of the intractability of manipulating experiments across trophic levels. And it's been done before in some pretty well-known experiments, but the large bulk of evidence we have really is single trophic level focused. So this is important because biodiversity is not only a single trophic level. We know that it transcends, both taxonomic groups as well as trophic levels. So, certainly plants are only one part of a very complex network of species interactions. And all of these different organisms are carrying out, vastly different functions. And, arguably they are all quite important for a range of different ecosystem services, or more recently named nature's contributions to people. And the linkages between, I guess, these species and the roles that they're carrying out, so the functions that they're carrying out, and ecosystem services are extremely complex as well. So there's really a lot of complexity that I think we need to get a handle on before we can sort of categorically say that increasing biodiversity increases functions and services, particularly that humans rely on. So I'd like to kind of now take a sort of turn toward I guess, the network approach that I'm particularly interested in. So, we know that, okay, biodiversity is changing, and it can change in different ways, as I said before. So imagine we've got baseline conditions here. We could have a turnover in diversity. So, hypothetically is the same number of species, but different composition. Or we might have diversity loss, and I guess the opposite to that would be that we can also have diversity gain. And these changes in biodiversity are inherently linked to changes in species interactions. And we know that the structure of those interactions and food webs or interaction networks is fundamental to determining the stability and species coexistence. But those interactions are also fundamental to the provisioning of ecosystem functions and services. So many of the functions that we typically tend to think about, such as predation, decomposition, etc., are a direct result of these species interactions between consumers and their resources, such that we can if we can quantify these interactions in terms of rates of, say, flux or matter, rates of energy flux or matter, we can look at these in terms of, for example, predation rates, herbivory rates, detritivity, or even direct uptake of nutrients. And these, depending on where they are in a food web or an ecological network, we can kind of map those onto different ecosystem services like pest control, pollination, etc. So if we are able to, I guess, quantify these fluxes of energy and matter through nodes in a food web, we're able to then basically extract that data to look at these biodiversity functioning relationships, but now in a multi-trophic context. And with the advantage that, ultimately we have a standardized measure with the same units across different trophic levels that we can then look at the relationship with diversity across a multitrophic system. So for example, like I showed before, we might have, say, decomposition, herbivory, and predation in a food web. We can then extract different facets of diversity from that food web, whether it's vertical richness, how many trophic levels there are, richness of nodes. If those nodes are aggregated, say species or individuals, we can look at taxon richness and regress that against, for example, energy fluxes at different trophic levels. And so that allows us to look at, for example, specific fluxes in response to changing consumer taxon diversity, like decomposition, herbivory, etc. And arguably, we can also combine those in different ways to look at metrics of so-called ecosystem multifunctionality, which I'm not going to go into really any more in this talk, but it's been used now quite widely actually in that context as well, for better or for worse. So before kind of moving on to a couple of case studies, I guess wanted to take the opportunity, being in a theoretical ecology seminar series, to actually explain kind of how we're actually going about the estimation, I guess, of energy fluxes in food webs. So we're using what we've, I guess, kind of called now the food web energetics approach. And I should just point out that there's, a number of different methods that are, I guess, kind of analogous to this one. So I'm not trying to say that, our approach is the best approach. And to be fair, we've kind of, I would say, assimilated the approach that's been developed by ecologists in the past, like John Moore and Peter de Ruyter, and sort of applied a metabolic perspective to that approach. So what we're looking at is trying to estimate the energy fluxes from one node to another. So imagine we've got a primary producer node, this plant here, and a herbivore, and we want to calculate the energy flux to this herbivore node. If we assume population equilibrium of this node, then we can assume that whatever energy is lost from that node has to be balanced by ingoing fluxes in order to maintain that equilibrium state. And so we've been doing this using I guess, relatively simple calculation whereby the energy flux is equal to the assimilation efficiency of that node. So what proportion of energy that it consumes actually gets converted to its own biomass. And that is then multiplied by the energetic losses. So in this case, we have a metabolic loss. So as organisms metabolize, they lose energy, and so that energy must be replaced by incoming energy in order to maintain their energetic balance, and also losses to predation, so to higher trophic levels. And so that's this X and L term here. So once we sum those losses together, we equal that with the incoming flux. And I just wanted to point out here as well that the metabolic losses, generally we're using quite, let's say, pretty generalized bioenergetic equations. So this is just, this one here is a classic metabolic theory of ecology, James Brown equation, whereby metabolism is scales with body size to this 3/4 power law and to Arrhenius temperature. Of course, there's a lot of contention over this 3/4 power law, and when we can, we're using more specific metabolic rate regression parameters that actually do vary quite a lot across different taxonomic groups. And of course, the loss term here is really just the sum of outgoing fluxes from that node that we're interested in. And in addition to that, like I said, we're accounting for assimilation efficiency as well. So, in addition to the balancing these outgoing fluxes, a consumer node also needs to balance the loss of energy that that consumes, which is not assimilated into its own biomass production. So that's this assimilation efficiency term, which this is just to show that that's also temperature dependent, and where we have information on resource stoichiometry, then you can also parameterize that based on, for example, carbon-nitrogen ratios. So the higher carbon to nitrogen ratio, the lower the assimilation efficiency typically of an organism. So with those energy fluxes, so that's been nicely implemented into the FluxWeb R package that Benoit Galzon has been really instrumental in making happen. Which has allowed us to really kind of expand this to highly complex systems. So we can look at, sort of fully resolved sort of species level food webs, such as this German soil beech forest food web. And once we calculate all these fluxes, so from resources to consumers, we can then sum those together for different sort of ecosystem functions that we might be interested in, such as rates of herbivory, carnivory, and detritivory. Okay. So I guess that's kind of the background. What I wanted to do then is move on to a current project that we have been working on, so quite a large number of us, where so I should also point out, I'm not really going in chronological order here, but probably more conceptual order. So I think, after a lot of the work that we've done with energy fluxes and food webs and trying to link that to to biodiversity and ecosystem functioning relationships, we've kind of come full circle to realize that we still haven't really targeted this underlying question that was, I suppose, one of the original driving questions that led to the development of this work. And that, we're trying to identify really what are the, I guess, aspects of food web structure. What is it about trophic interactions that actually potentially modulates the relationship between biodiversity and ecosystem functioning? And so over the last few years now. This is a project that a large group of us have been working on, where we've been trying to compile food web data sets from wherever we can that has the kind of the essential ingredients to calculate energy fluxes, to try and tease apart what some of these mechanisms, the food web mechanisms, might actually be that modulate the biodiversity functioning relationship. So you can imagine we have, say, a set of communities or food webs, and we can regress their taxonomic richness with ecosystem functioning. And so the classical expectation would be that we might have this sort of saturating relationship with increasing diversity and increase in ecosystem functioning. And if we just focus on a couple of these food webs, so taking a look at this sort of hypothetical low diversity food web here, we, of course, can calculate rates of predation and primary consumption. So imagine this has a species richness of 6. And we can also then calculate, a range of different food web properties to try and describe why changing richness might influence rates of energy flux. And so we've thought about this for quite a while, but we've kind of zeroed in on two major I guess two key components of food web structure to try and help understand this kind of underlying relationship. So the first one is vertical diversity. This was published a few years back by Xiaoping Wang, who actually I think is going to be presenting a bit later in this series. So this nice theoretical study looks at how increasing maximum trophic level should lead to increases in primary production in food webs, basically through losses of energy, metabolism, and feeding efficiencies, needing to be replaced through nutrient uptake at the bottom of the food web. So we decided to try and apply this to a large set of empirical food webs. So we're looking at maximum trophic level as an estimate of vertical diversity. And the other hypothesis that we're looking at is that trophic complementarity, which was, I suppose, developed or named, if anything, by Timothy Poissois, whereby sort of increasing partitioning of resources among consumers should lead to a greater total uptake of energy and matter in the system, which is essentially just a multitrophic analog of so-called niche complementarity, which is one of the original mechanisms that was kind of identified in these sort of early biodiversity ecosystem functioning experiments. So this is a situation where we've got high niche complementarity between predators and also between primary consumers. And so the more dissimilar species are essentially in the resources that they feed on, the greater total uptake of resources we should see in that system. And so the greater total rate of ecosystem functioning because of reduced competition. So the thing is, this was never really developed for unipartite networks, so food webs. So in this paper that was published by Timothy Poissois in 2013, he was looking at bipartite networks, so sort of like a plant pollinator system, and using nestedness to kind of approximate this this metric, trophic complementarity. And so we're now taking a different approach to that by measuring trophic dissimilarity based on the Jacquard's dissimilarity of species in networks in terms of their feeding interactions with resources as a sort of unipartite analog to this metric. So we then might expect, if we assume these hypotheses actually play out in empirical systems, we might expect that with increasing species richness, we also see an increase in vertical diversity of a food web. So the more diverse a food web, the more trophic levels they tend to have. And as a result, we might then see this vertical diversity hypothesis play out, whereby more trophic levels equal more functions at different trophic levels. And alternatively, or in conjunction, we might also see increases in trophic complementarity, so a sort of diversification of the resources that species feed on as they kind of partition resources in more diverse food webs. Particularly as there's greater resource availability, we might expect greater, partitioning of those resources among consumers, and again, that hypothetically would lead to an increased rate of energy fluxes to those food webs through reduced competition. So, just quickly, this is sort of an overview of the data set that we're now working with. And believe me, the irony is not lost on me that I'm speaking in a seminar series for the Asia Pacific region, and that is cut off from this map. It's almost criminal that I've done this, but ultimately, this is where the data come from that we're able to access. So unfortunately, it was a bit sparse over on this side of the planet. Hint to anyone interested in working on food webs, we need more food web data that has both body size and biomass information in the same data set. So with that apology aside, we've got now about 320 food webs, I think, that we're working with across 4 different systems, so marine, soil, stream, and lake ecosystems. And just as you can see from some of these example graphs here of different food webs, there's a massive variation in diversity and structure of these food webs. And what we're looking at is how, so using a structural equation modeling approach, we're looking at how taxon richness, so the diversity of these food webs, influences their maximum trophic level and consumer dissimilarity, trophic dissimilarity, and how that in turn leads to changes in primary production and predation. And presumably we would see positive effects, except for maybe maximum trophic level, which might actually have a negative effect on primary consumption through top-down control. So what did we find? So these, it's still a work in progress, but overall we found, pretty clear, I would say, with a sort of guarded voice relationship between taxon richness and both primary consumption and predation in these food webs. But what really jumps out here is just a massive amount of variation around those relationships. And so this is looking across all ecosystem types. So we've just combined it all together. And so, not surprising, there's a huge amount of variation here. So this, excuse me, this is the structural equation model that we've analyzed across all ecosystem types. And what we tend to see is a consistently positive effect of taxon richness on both primary consumption and predation rates. But when we look across food webs, the only real sort of food web mechanism that we see clearly playing out here is maximum trophic level. And, with a very clear positive effect on predation rates. So the more trophic levels there are, the greater rates of predation we see in food webs, which I suppose is not too surprising. And what we can see here is that sort of a decent proportion of the effect of taxon richness on predation is indirect via this effect on positive effect on maximum trophic level. So food webs with more species tend to have a higher trophic level, maximum trophic level, so greater vertical diversity, and that tends to lead to significantly increased predation rates. So we also looked at this within ecosystem types, and there's again just sort of example food webs from across those ecosystem types, which they are really highly variable, and I think that in itself is quite interesting. But I'm again not going to go into that here. There are really fundamental biological differences between, for example, soil food webs and aquatic food webs. But again, by and large, we see pretty clear positive relationships between diversity and function, with the exception of primary consumption in lakes. But when we then model that again using the structural equation models, we can see that even for lakes where we didn't find a significant effect of taxon richness on either of those functions, once we actually incorporate these food web metrics or food web properties into this model, it actually kind of reveals that there are, in fact, significant effects of diversity on functioning, but it's highly dependent on the structure of the food web. So, for example, maximum trophic level has a pretty strong negative effect on primary consumption, so probably through these through top-down control, and at the same time, pretty clear positive effects on predation through increases in maximum trophic level and predator dissimilarity or trophic complementarity. And that pattern here we see pretty clearly in marine food webs as well. and sort of a little bit of variation across streams and soils, but the story tends to hold pretty true that diversity, by and large, has an indirect positive effect on these functions via some or all of these sort of food web properties. So, we feel like this is reasonably good evidence that the structure of food webs, this multitrophic structure, is pretty important for modulating this relationship between biodiversity and ecosystem functioning. So hopefully, this will help to convince people that sort of multitrophic structure of systems shouldn't really be ignored or kind of tried to, explained away. It's really a kind of key component to understanding this effect that biodiversity has on ecosystem functioning. Okay, so I kind of want to switch gears a little bit here and focus in on some more, I guess, kind of specific mechanisms that chronologically, we're going back in time a little bit here, but this was quite a fun project that I worked on a couple of years back, where we wanted to look at how some sort of classic food web theory, so looking at top-down and bottom-up control, might help to explain ecosystem functioning along sort of classic environmental gradients of plant diversity. And this idea came from, I guess, looking at a couple of different studies. So, there's considerable evidence that increasing plant diversity leads to increases in top-down pressure from natural enemies. So, particularly in cropping systems, this is very commonly studied. How do predators control insect pests, particularly herbivores? And this has been shown to clearly increase with plant diversity. And the other side of this is that increasing plant diversity might actually limit the performance of herbivores. And that kind of got us thinking, well, what if this limitation, this bottom-up limitation, combined with this top-down limitation, in fact, sort of work in concert to control the impacts of herbivores on plants in higher diverse systems. So this is essentially what we did. We proposed that with increasing plant diversity, we should expect to see declining bottom-up effects. So these bottom-up effects are expressed as a positive value, and that it's the resource or energy gained by herbivores from their plant resources. And the opposite should be true for top-down effects, whereby increasing plant diversity creates more, habitat, niche diversity for predators, which at the same time impose a top-down pressure or an increased top-down pressure on herbivores at high plant diversity. And so if we sort of look at both sides of this together, we should see a sort of net increase in total herbivore control with increasing plant diversity. And these two aspects of herbivore control have actually been hypothesized sort of in isolation of each other. And actually, this has been looked at before in these biodiversity functioning experiments, but never really in concert with each other. So, which is kind of interesting because top-down and bottom-up effects are really indistinguishable from each other. I mean, they occur simultaneously. So to look at one or the other is kind of putting blinders on in a sense. And as a result, we would expect then that the impacts that herbivores have on their plants that they eat should also decrease with increasing plant diversity if we do actually see this increasing net control of herbivores. So we looked at so first of all, what we had to do is kind of come up with a way of quantifying these top-down and bottom-up effects, and essentially used this energy flux approach that I've already presented to calculate these top-down and bottom-up effects, which have been used quite a bit before in past studies to quantify food web stability. So it's kind of just taking those parameters and individually looking at those top-down and bottom-up effects. So if we're looking at the sort of multitrophic control of herbivores, we basically looking at the top-down effects, so the effect of the predators of that herbivore on this population, which we just simply calculated as the total outgoing flux to their predators per unit biomass of that herbivore population. And nearly the opposite for bottom-up effects. So the total ingoing flux of energy to that herbivore population per unit biomass multiplied by their assimilation efficiency to account for the fact that they're not actually assimilating 100% of the energy that they are consuming from these plants. And so basically, this is just synthesized from 2 years of data that's been collected from the Jena experiment, which is a pretty well-known plant diversity experiment in Jena, Germany, and also the Cedar Creek experiment that was originally set up by David Tillman in the early 90s. And so we had reasonably comparable data from these two different pretty large-scale field experiments. And calculated these top-down and bottom-up effects. And lo and behold, we found, some evidence that we have a declining sort of bottom-up effect of plants on their herbivores. And arguably the mechanism behind this could be that the more diverse plant communities are, the more diluted the resources are for herbivore specialists, and so this should, well, theoretically, that should limit population growth of particularly of specialist herbivores. And on the other hand, we've got increasing habitat diversity for predators, and so greater predator biomass production and consumption of herbivores. When we then looked at the log ratio of those two variables, we found that there was actually a significant increase in the sort of net herbivore control metric that we calculated. So that seemed to kind of fall in line more or less with what our hypotheses were. And interestingly, when we then calculated the top-down effect of herbivores on plants, using a similar formula, so just the total flux from plants to their herbivores per unit biomass of plants, we found a significant decrease of herbivore effects in the highest diversity plots. So the greater the plant diversity, the stronger those sort of bottom-up and top-down controls seem to be, and that could be what's leading to this sort of declining impact that herbivores have per unit biomass on plants, which I should point out is somewhat contradictory to previous results of past studies looking at how herbivores respond to this plant diversity gradient. So there are actually higher numbers of herbivores, and we also calculated total fluxes. So the total energy flux to herbivores is in fact higher in these higher diversity treatments. But per unit biomass of plants, their effects on those plants is actually lower, because there's also much greater plant biomass production in those high diversity plots. Okay, so hopefully I haven't gone too much overtime here, but just in summary, hopefully I've sort of convinced some of you anyway that it's quite critical that we're incorporating information on species interactions to really understand what's going on with biodiversity functioning relationships. And in the past, these have been either ignored or intentionally excluded from experiments. But I think it's high time that we really start trying to very specifically tackle this problem to better understand, in real ecosystems, why we see so much variation in the relationship between biodiversity and ecosystem functioning. So that might help to kind of reconcile some of that variation that we see. And yeah, I mean, we do see some consistencies across ecosystem types, but also a lot of differences. And actually, I think much of the interesting sort of results that we might be able to start finding are really coming from those differences among ecosystem types. And that's, I think, somewhere that would be quite interesting to start looking at next. I would say that this is an argument for food web metrics potentially being useful. I know that, sometimes food web metrics can be a bit vague, and it's hard to understand kind of, what they're really telling us about a system. But I think if we're kind of careful about which metrics we're using and have, good sort of mechanistic basis for those metrics, they can provide quite a lot of insight into what's going on. And I think that there's going to be a lot of other metrics that are probably as or potentially more useful than what we've found so far to help kind of further kind of disentangle what's going on with these relationships between diversity and functioning in multi-trophic systems.